My pleasure and honor to introduce Dr. Geraldine Seydoux. I have to confess, to prepare this introduction, I tried to summarize Dr. Seydoux's work on RNA granules and condensates onto one page of paper, and it soon phase separated into like 20 pages. So here I gave you a condensate, or I mean a condensed version of my introduction. So Dr. Seydoux finished her undergrad study in biochemistry at the University of Maine in 1986 and obtained her PhD in molecular biology in 1991 at Princeton University in the lab of Eva Greenwald, where she studied cell-to-cell interactions in C. elegans embryos. And for her postdoctoral training, Dr. Seydoux joined Andy Fair's lab, where she studied the distinction between somatic and germline cells during early C. elegans development, particularly in relation to RNA transcription. In 1995, Dr. Seydoux joined the faculty in the Department of Molecular Biology and Genetics at the Johns Hopkins University School of Medicine. And she is currently a professor and holds the title of Huntington Sheldon Professor in Medical Discovery. Dr. Seydoux is also a Howard Hughes Medical Institute investigator. In addition, she serves as the Vice Dean for Basic Research at the Johns Hopkins University School of Medicine. Dr. Seydoux is a distinguished leader in developmental biology. Her work has fundamentally reshaped our understanding of early embryogenesis and the formation of the germline. Using C. elegans, her research investigates how a single fertilized egg develops into a complex organism with distinct somatic and germline tissues. Just to give a few examples of her very broad research achievements, Dr. Seydoux is particularly renowned for her finding that the global inhibition of mRNA transcription is an essential first step in establishing the embryonic germline. And she has further elucidated the post-transcriptional mechanisms that regulate gene expression and promote germ cell fate. Her work also showed how localized changes in protein diffusion drive the formation of crucial protein gradients within the cytoplasm, a key process in establishing embryonic polarity. Furthermore, her lab identified a novel class of intrinsically disordered proteins that coat and stabilize the surface of RNA granules, demonstrating for the first time an intracellular pickering agent. Dr. Seydoux has been recognized with numerous honors. For example, in 2001, she was awarded the MacArthur Fellowship, and in 2002, Discovery magazine recognized her as one of the 50 most important women in science. And she was elected to the American Academy of Arts and Sciences in 2013 and to the National Academy of Sciences in 2016. In 2022, she received the Gruber Genetics Prize. But beyond her brilliance as a scientist, I want to mention that Dr. Seydoux is also widely admired for her clarity of thought, her humility, and her generosity as a mentor. Her work continues to inspire new generations of researchers in RNA biology, developmental genetics, developmental biology, and beyond. So please join me in welcoming Dr. Geraldine Seydoux.
Wow, that was very, very nice. Thank you. All right, well, thank you so much for that wonderful introduction, and thank you all for being here. We're going to talk today about condensates. I also, of course, want to thank the organizers of this meeting for bringing me in, as I'm very much an outsider. I've never worked on splicing in my life, but I've learned a lot in this meeting, and I really appreciate how easy it's been to have wonderful conversations with many of you already. So today we're going to talk about condensates, and in particular I'd like to talk about their involvement in mRNA decay and translational regulation, and we're going to focus on condensates in the cytoplasm. But as you know, biomolecular condensates have been implicated in many different steps of the RNA life cycle. And I've just highlighted some condensates here, starting with transcription, splicing, exit through the nuclear pore, and then several condensates in the cytoplasm that are thought to perhaps regulate storage of mRNAs or decay of mRNAs. And then there are some condensates that are specific to particular cell types. It is thought that several of these condensates form by phase separation, which is a concentration-dependent process whereby molecules that interact with one another when they're at a low concentration can be nicely dissolved in the cytoplasm, but when they exceed their critical concentration will form condensates that demix from the cytoplasm. And it's often assumed that some kind of activity happens within the condensates. But you have to remember that because this is a concentration-dependent process and these condensates form within a cytoplasm where many of the same molecules that are here are also still in the dilute phase. And so a big question in the field often is, where is the activity? Is the activity really in the condensate only, or is it still in the cytoplasm where many of these molecules are? And that's the question we're going to address today. So we're using C. elegans as our model system, and that's because C. elegans has a very nice early invariant lineage. So here you're looking at pictures of a C. elegans embryo doing all of its division all the way to making a little worm. And here you're looking at a schematic of these divisions. And what you might notice is that the divisions are asymmetric. You get a big cell and a small cell, big cell and small cell. And these asymmetric divisions distinguish the cells that will give rise to the germline from the cells that will give rise to the body of the worm, as shown here. And we know that the cells not only are different sizes, but they inherit different maternal proteins. So when I say maternal proteins or maternal RNAs, I'm talking about RNAs that were synthesized during oogenesis and that are present in the fertilized egg, the zygote. And these proteins are not just uniformly distributed to all of the cells. Some of them become segregated to the small cells, other to the large cells. And in particular, many years ago, we showed that proteins that inhibit transcription go to this side, and proteins that activate mRNA decay go to this side. And as a result, you have different patterns of transcription. So transcription is off in these cells. Eventually, it comes on. And it's on in all these somatic cells. So that's, if you look at zygotic mRNAs, that's the pattern you would see. But if you look at a maternal mRNA, you see the opposite pattern, where the maternal RNA is preserved in the germ lineage and it's actively degraded in the somatic lineages. So this offers a very nice model to study mRNA decay, because the embryos are always developing in exactly the same way. So if you look at a 4-cell embryo, you know that mRNA decay has started in this cell, but not in that cell yet. And we can also study translational control, because these maternal mRNAs, each of them exhibit different translational patterns, and I'll show you that. So the other thing that's very interesting is that RNAs, the maternal RNAs, go into condensates. So here is an in situ hybridization where we're using a little probe that recognizes this splice leader. In C. elegans, 70% of mRNAs are transpliced and receive a 20 or so nucleotide sequence. And so when we do in situs to that sequence, we see all, or 70% of mRNAs. And you can see that there are lots of mRNAs in the cytoplasm, of course, but you see these nice granules both in the somatic cells and in the germ cell right here. If we do the same experiment using an oligo-DT probe to specifically look at messenger RNAs, we see a different pattern. We see that there are now no condensates in the somatic cells, but you still see the condensates in the germ cells. So there are at least two condensate types, some that enrich deadenylated RNAs and some that are polyadenylated. And it turns out that we know a lot about the proteins that are in these condensates. So the ones that are in the soma are traditional P bodies or processing bodies. These are condensates that enrich the mRNA decapping machinery, and they and also different kinds of scaffolds that bring all of these proteins together to initiate mRNA decay for deadenylated transcripts. In the germ granules, there's actually more than one condensate. There is these, we call them arrested P bodies, where you don't have the full decapping complex and the mRNAs still have poly-A tails. And then there's an even bigger, more prominent granule called the P granule, where actually the majority of poly-mRNAs end up there. And I'll discuss these more in a minute. So the question is, what are these condensates for? Are the P bodies important for decay? Are these somehow regulating translation? And the way that we're going to address this question is by examining scaffold mutants. And what I mean, we have, the community of the P body community has identified proteins which are not enzymes in the P bodies, but they're scaffolds of the P bodies. So we can get rid of those. And we have identified scaffolds for the P granules, so we can do the same kind of analysis. And all of the work that I'm going to talk about today is unpublished, and the P-body work is by Brennan Dunlofsky, a graduate student in the lab, and the germ granule work is by Will Simmons. So let's talk with the P-body work. So just to give you a little bit of background here, P-bodies were initially proposed to be sites of messenger RNA decay. Then later on, they were thought to be more important perhaps for storage. And so the debate is still on, exactly are they doing decay, storage, but there's also papers out there that said that they're doing neither of those things, okay? So we're gonna look at that in C. elegans, and as I said, we're gonna focus on scaffold mutants. So here's an example of a scaffold mutant. So EDC4 is a protein that links the decapping complex to XRN1, and in wild type, again, the decapping complex is present in these P bodies, and also we see XRN1. When we get rid of EDC4, we still see decap1 in P bodies, but now these enzymes that are critical for decapping and RNA decay are no longer enriched in the P bodies. They're still present, they're all over the cytoplasm, they just don't enrich in the P bodies. So we can ask, well, what happens to mRNA decay in these embryos? And here is mRNA-seq data set where we collected 2-cell embryos, 8-cell embryos, and 24-cell embryos, and we look at all the transcripts present in those embryos, and we can detect about 2,000 mRNAs that decay during this period. So this is in the wild-type situation. So now we're going to do the same analysis on these EDC4 mutants, and what we see is that most of the mRNAs are unaffected. Okay? There are a few mRNAs that show slower decay. There's some that actually show faster decay, but by far, most mRNAs we see no difference whatsoever. So let me just show you what I mean. Here is a typical mRNA that undergoes decay. So this is now we're doing single molecule fish to this particular maternal RNA. It's present in a 2-cell and it gets degraded. In this 8-cell stage, which is kind of the intermediate on the way to degradation, we see the RNA clustering in these little patches. Of course, it's also going to germ granules over there, but don't look at those. We'll talk about those later. We're talking about the ones that form in the somatic cells where the RNA is degraded. And these granules are the P bodies. So this mRNA clusters in the P bodies. But of course, when you look at EDC3-GFP, yes, it's in the P bodies, but it's also all over the cytoplasm, and so is this RNA. This RNA, you can still see pink little dots that are not in the green EDC3, okay? So, but nevertheless, this mRNA enriches in P bodies, And when we look at these scaffold mutants, either the EDC4 one, which I already talked to you about, or this other scaffold situation, we now reduce the abundance of this mRNA in the granules. So in the 8-cell stage, about 25% of the mRNA molecules are in these clusters, and this is reduced in these two mutants. However, degradation doesn't change at all. And in fact, we can see this with mRNA-seq, specifically looking at the spin-4 behavior in wild type and these two different mutants, there's no difference. So that says that mRNA enrichment in P-bodies, or the enrichment of DCP2 and XRN1, is not required for most mRNA decay in early embryos. And so we think that P-bodies assemble simply because when there's a lot of mRNA decay that happens, you make a lot of these decapping RNPs, they exceed their critical concentration, and they saturate the cytoplasm. But the mRNA decay is mostly going on in the cytoplasm. So we think P-bodies are not essential for mRNA decay, and they might be an example of incidental condensates. Condensates that have no function, they're tolerated by cells. They're still useful as markers of a lot of mRNA degradation is happening in that cell, but nothing special is happening on there. All right, so let's move on to the germ granules. So the germ granules are specific to this lineage where maternal mRNAs are stabilized. And the germ granules have been observed not just in C. elegans, but in many different animals, and they always segregate with the germline, which is why they're called germ granules. And they have been proposed to do many different things. Most recently, they've been proposed to regulate mRNA translation, both by repressing translation of mRNAs that are in the granule and also activating the translation of mRNAs that are on the surface of the granules. Okay, so that's what's out in the literature. So let's take a look at that. So here, my lab had identified these intrinsically disordered proteins, which when you get rid of them, you now reduce the poly-A accumulation in germ granules. You still have these little P-bodies out there, but now these polyadenylated large granules are reduced. What is MEG3 and its paralog MEG4? They are intrinsically disordered protein. They're highly basic, so they bind mRNA sequence non-specifically. And if you mix MEG3 protein with purified RNA, you get these little clusters, these little hard aggregates of protein and RNA. And when we asked, well, in embryos, what is it that MEG3 binds to? This was work by Sean Lee when he was a postdoc in the lab. He did an iClip of MEG3. He identified about 500 mRNAs. And these mRNAs, what they had in common was that they were less translated than your average embryonic mRNA. So MEG3 favors low translation mRNAs. It seems to bind all over the mRNA. So these are the footprints of where MEG3 binds in a metagene analysis, and you can see it binds all over the open reading frame. It also binds on the UTRs. And then when we do a mass spec on what kinds of proteins does MEG3 interact with, it interacts with proteins that would be thought to be at the cap, in the UTR, in the poly-A tail, but also in the body of the transcripts. And many of these proteins are translational repressors. So this is all consistent. It looks like MEG3 is just binding to any mRNA that's not engaged with ribosomes in the cytoplasm. Okay, so let's ask now, is this important? Do we care that MEG3 makes these condensates? So here's a typical mRNA. So this is a maternal mRNA. It is present in the 2-cell stage, and then it's eventually degraded in the soma, and so you can see it only in this cell at this stage. And it's in the granules, you can see in the granules. Now, you'll appreciate that there's a lot of mRNA also in the cytoplasm. This is true to every mRNA that we've seen localized to the granules. We've actually done in situs to over 400 mRNAs that bind to MEG3. 90% of them are enriched in the granules, but none of them are 100% in the granules. There's lots in the cytoplasm as well. Okay, so now we can ask, what happens when you get rid of MEG3 and its parallel MEG4? Well, now this mRNA is nicely uniformly distributed, okay? But you can see it's still degraded in the soma and maintained only in the germline. But you'll notice there's a little bit less RNA in this germ cell than in this one, and that's just because there's now no preferential enrichment of this mRNA in these cells that are getting smaller and smaller. So really, this shows you that the importance of these granules is to localize mRNAs to the right place. Because if you don't have the granules, then the RNA are just distributed evenly to all of the cells, and you just get an equal amount. Of course, the RNA is degraded in the soma, but not in the germline. But here, you're really enriching the RNA in this cell. Okay, so that's good. That tells us the granules are important for at least one thing, localizing the RNA. What about translation? So ZIF1, now we're looking at the protein. So remember, this is an RNA that's present in the two-cell stage, in both cells, but it's translated only in the AB cell, this somatic cell here. So there's RNA here, but no protein. And this continues. It's not translated in any of these cells here. Eventually, it gets translated in this cell down here. You get a little bit, the start of translation here with a little bit of pink protein. So now we want to know, well, does it matter, perhaps, the granules help keep the mRNA off in these blastomeres? And so we can ask, in a MEG3/4, where the RNA is no longer in the granules, is there any translational defect? And there isn't. The RNA is still not translated in this cell, and it's still not translated here, and it's also activated in this germline founder cell at the right time. Okay? So this was perplexing because I told you that people had, looking at germ granules in Drosophila, suggested that germ granules might be involved in repressing translation early and allowing for the translation activation to happen at a specific stage. And here we're not seeing any effect. Okay. So we decided to take a closer look and to really visualize translating mRNAs, to ask where is mRNA translation happening. And we're using a method, this is in collaboration with Eric Griffin, who used to be a postdoc in my lab and now, of course, has had his own lab for many years, but he has applied this technology that's related to the SUNTAG technology that some of you might have heard about, where you put multiple epitope tags next to the ATG of your favorite gene. And then the idea is that when this RNA is translated, these epitopes are the first to come out of the ribosome channel. And in a polysome situation like this, you're going to have lots of HA epitopes on one mRNA, and you can use antibodies against HA to visualize this. And you can visualize this live using a nanobody that is expressed in the embryo and that is tagged with GFP, or you can do this fixed using just a regular anti-HA antibody, and then you can also use probes to see the mRNA. So I'm going to show you examples of both of that. So let's start with the live situation. So this is what you see. This is a live embryo with the anti-HA nanobody, and the HA epitope is on this ZIF1 mRNA. And remember, the mRNA is everywhere. You can't see it here. We don't have a way to look at the RNA live yet. But you can see all of these beautiful little dots here. These are the translation sites. You also see fluorescence everywhere. That's the free nanobody that likes to go into nuclei. But you can see the high concentration of nanobody on translating mRNAs. So you see the beautiful specificity of translation, where it's only happening in this anterior cell. So we wanted to try to get really quantitative about this, and it's a lot easier to be quantitative when we can see both the RNA and the protein. So now we're going to do the same experiment, but fixed, okay? So this is still a 2-cell embryo. This time I'm showing you the RNA, just by single molecule fish. You can see the RNA nicely all over. This is a thin Z slice. That's why it's not so crowded. You can see individual RNA molecules. There's a little bit, this stage, the germ granules are still forming, but you can see little clusters of mRNAs out there in this cell, but not in this cell, because the germ granules don't form in that cell. So now let's look at the protein in pink. So here we see the protein, and we can co-localize the RNA and the protein, which will give you little white dots. And here's a close-up, so an example where you see a bunch of RNA molecules here. And here's the protein. And you can see that only some mRNAs are being translated. Not all of them. Or at least not all mRNAs are translated with sufficient number of ribosomes for us to see the nascent protein. Okay? And then on this side, RNAs are not translated. And here's there's a granule marker here. Here's some RNAs on the granules, and they're not translated. But even an RNA that's away from the granule is not translated. So now, with this approach, we can be very quantitative, and we can systematically count how many RNA molecules are there, how many are associated with protein signal, and that tells you the percent of the mRNA that are translated. So here's an example where I'm going to express these data as percentages. So here is in this AB cell here, there's hundreds of mRNAs in this AB cell as we go through the sections, okay? And about 50% co-localize with protein. So about 50% of the mRNAs are translated. In the P1 cell over here, there's also lots of mRNAs. Most of them are in the cytoplasm. A few, as I said, are in the granules. At this stage, the granules are just forming, so they're small. So here is the RNA in the granules. But none of these RNA molecules are translating, okay? Because there's no the hatch bars here show you the proportion of translating mRNAs. So now, let's look in a mutant where these little blue granules are not going to be made. So that's our MEG3, MEG4 mutant. And basically, it's exactly the same. The only difference now is there's no granules, or barely any, so there's very little RNA that co-localize where our granule marker. But you can see, we still see nice translation in the AB cell, 50%, that's all good, and no translation at all there. Okay, so again, the RNAs don't need to be in the granules, or the granules don't need to be around for anything for translational repression. What about translational activation? Because eventually, this cell is going to start turning on the translation of this mRNA. And this is a cell that has lots of germ granules, which are shown in blue here. So again, the RNA, and some of the RNA is in the granules, and some of the mRNA is out there. And then here we're again looking at where is the protein appearing. And there we saw something interesting where, so here the data, I'm actually showing you actual raw counts, just to show you how amazingly reproducible this is. Each line here is an embryo, and it's one P4 cell. And you can see that this is the total number of RNAs that are outside of the granules and the total in the granules. So it's around 100 molecules that we can detect at this stage. And in the little darker dots show you the number of mRNAs that are associated with protein. So in this particular cell, you see that, I don't know, about 25% of the mRNAs are translating, and so on. So we see very nice levels of translation in mRNAs that are out there, away from the granules. Inside the granules, we have lots of RNAs in the granules, and we don't see any translation. If we do, these little dots here represent 1 or 2, and those mRNAs, they could be on the surface of the granule, for all we know. It's actually very hard for us to tell whether they're inside or on the surface, okay? But it's very little. It's not as many as we see on the outside. So there's something funny going on with the granules where if you're inside the granule, you don't get to be translated, okay? And then, of course, we did it again in this MEG3/4 situation where now the RNAs are not recruited efficiently to the granules, so there's very little RNA in the granules, but we still see very nice translation in the cytoplasm. There's less RNA overall, because remember, the granules are required to enrich the RNA to the right cell. So that's why you have about half the amount of mRNA, but that mRNA is still translated just fine. Okay, so what is going on here? Just going back to this was surprising to us. We thought, okay, if the granules are not required for anything, maybe RNAs could be even translated inside, but they don't seem to be, okay? And this is, I'm showing you just one mRNA here, but we've done this for other mRNAs, and this seems to be the recurring theme, that mRNAs inside the granules are not translated. Why? And so then we asked a really kind of simple question, which is, where are the ribosomes in all this? So this is RPS6, a ribosomal protein tagged here with mCherry. So it's all over the cytoplasm. These big circles here are the nuclei, so that's good. We don't have lots of ribosomal proteins accumulating in the nuclei. They're all out there in the cytoplasm. And here's a funny looking nucleus. I blow it up for you here. This is the germline founder cell, and at this stage, the granules are nice and big, which helps us to see them. They're also perinuclear, and you'll notice that the ribosomes are excluded from these granules. So that explains why we don't have translation inside the granules. There's no ribosomes. So the granules are condensates that somehow can exclude ribosomes and create an environment where mRNAs are not translated. So I'm going to end way ahead of time, so you'll be glad, Shalini, because I know you were worried about the timing and everything. So it's all good. The take home here is that germ granules have these scaffolds that recruit poly A plus mRNAs. I should mention that I didn't talk about these, but there's also these arrested P bodies that associate with these P granules. We only, in these experiments that I told you about, we only changed the fact that the enrichment of the poly A mRNA in these granules. And these granules are formed by this intrinsically disordered MEG3, which condenses low translation mRNAs in these granules, and they exclude ribosomes. And recruitment into the granules is not important for translational repression or activation. The mRNAs are going to know, probably through proteins that bind to their 3' UTR, when they need to be translationally activated or translationally repressed, and that's all decided in the cytoplasm. Okay? However, this condensation mechanism is really good to enrich RNAs to this cell, which, remember, there is no transcription in this lineage, so you need to hold on to as much maternal RNA as you can in these cells, and this is the mechanism. So you really gather up your mRNAs and make sure that they are there, but this is not regulating translation. It's just regulating localization. And then just to summarize again what we said about P-bodies, P-bodies enrich decapping enzymes and deadenylated mRNAs that are on their way to be degraded. And we think they formed simply because there's saturation of the cytoplasm by these decapping RNPs, and enrichment of mRNAs in these P-bodies or the decay enzyme. I showed you an example where XRN1 and DCP2 were missing from the P-bodies. Really is not important. Most mRNAs are decayed just fine. And so we think this is an example of incidental condensates. So this is all I wanted to share with you today. This was really meant to be just something for you to think about, because I know whatever part of the RNA life cycle you study, you have been told that there are condensates that do all kinds of amazing things. It's very important to look at the data carefully. If people are mutating a decapping enzyme and then saying that this has an effect on P-bodies and on mRNA decay, that's exactly what you would expect for an incidental condensate. If you don't have the enzyme to create the RNP is not going to phase separate, and also the RNA is not going to be degraded. But these are correlation, not causation. And so really P-bodies, we still don't know whether maybe in some contexts they might have a function, but at least for now, our working model is these are incidental condensates, tolerated by cells, but not functional. Of course, evolution can see a good thing when it sees it. Some condensates are useful. In this case, the germ granules are an elegant mechanism to enrich maternal RNAs to cells that are not transcribing, which need these maternal RNAs to start translating proteins, etc., to differentiate these germ cells from the somatic cells. But again, this is not important for regulating translation. Regulation of translation is happening at the level of RNPs through proteins that bind to the UTR and do deadenylation, as we've heard in many of the wonderful talks that we heard this morning, for example. So here we have an RNA localization function, but not translation. So this is all I wanted to say. This was the work of two graduate students, Will Simmons on the germ granule side and Brennan Donlowski with the P-body side, and I look forward to your questions. Thank you.
Geraldine's beautiful demonstration. Obviously, P-body is for RNA storage, not for degradation. In your system, it's clear. I get a little bit confused with your germ granules in the P-bodies and also your P granules. How can we define this?
How do you define?
Do we have a specific marker?
Oh yes, so, and I did not go into that, but we have looked at many different proteins. So the typical P-body proteins are in these arrested P-bodies. We can see them localized next to the P granules. So just by microscopy, we can distinguish between these two condensates. Yes. Yeah. So the condensates are defined by their composition, basically. And most of the mRNAs are in the P granule, okay? Yes.
So we use the PABPC1 standing for the P-body in the mammalian cells.
Sorry?
We use a PABPC1 as a marker.
Yes.
For the P-body.
Yeah.
I don't know whether that also including the P granules in your system.
Well, P granules, it's possible, I guess, but P-body markers that we've looked at are not in the P granule. They're in these adjacent condensates, which are these arrested, what we call arrested P-bodies, because they don't have all the complement on the enzymes for decay. Yes.
Okay, thank you.
Yeah, Geraldine, that was really great. Is there a mechanism that segregates the MEG3 or the granules into the germline cells?
Yes, yes, there is. So this is what we studied before getting into all this, is is there asymmetric segregation of these proteins? And it happens by regulated diffusion. So in the zygote, there are kinases that segregate to the cortex on one side or the other. And these kinases can modulate the diffusion rate of proteins, and that creates concentration gradients. And MEG3 is one of these proteins that becomes enriched in the posterior because its diffusion is slower in the posterior, and it stays there. And then it condenses, picks up the RNAs, and then makes these granules.
And that can be sustained through cell division?
Yes, every cell division. This happens, so that's why you end up really concentrating the RNAs in the last cell. Yes?
Really beautiful work. And I love your conclusion about the P-bodies not being important for most mRNA decay, but you showed about 10%.
I knew somebody was going to like, yeah, what about those 60 or so?
So
Yes.
do they have a feature that there's something about them that is enhanced by being in the condensate, or is
absolutely.
that just a total secondary effect?
And really, so this is the data that I showed you is in the last month, where we found that, okay, most mRNAs don't care, but they are a subset that do care, okay? And so now we need to understand what's going on with these mRNAs, because when you do mRNA-seq, of course, we don't really know what's wrong with these mRNAs. Are they not decayed right? Or are they starting to be transcribed more? Because this is in cells that are transcriptionally active. And in fact, when I look at what are these mRNAs, what do they code for? It's all the proteins you guys study. It's RNA metabolism. There are some splicing factors in there. And so I'm thinking that there is, remember, when these mutants, we got rid of EDC4 or EDC3. And so it could be that there is some kind of stress response, and we're going to follow that. So right now, I don't know whether these mRNAs are really not degraded properly, or there's some other stress response of the cell. Yes?
Yeah. So Geraldine, here. Yeah, beautiful data. So 2 questions. One question is, what is special of the granule, of the germ granules that is required for localization?
For localization, as I was mentioning, it's really MEG3 protein, even in its diffuse form, is enriched in the posterior. And then it condenses there because there's a higher concentration of it there.
And what happens to the mRNAs after they get there?
Yes, so the mRNAs do seem to be able to get out of these granules, in particular when they become translated. And that's because we think that perhaps these granules are actually more dynamic than just you could see in fixed pictures. We don't have a way right now to look at the RNA live, but it does seem that when an RNA is translated, it seems to come out of the granule, perhaps because it was always coming out and coming back in, but when it gets cut by a ribosome, it stays out there.
Good stuff. My question is, how do you know that the ribosomes are excluded? Is it possible that the ribosomes go in there, but there's something preventing them from recognizing the mRNAs and initiating
You're right.
translation?
Why are they not in there?
Maybe they're not retained in there. You could.
I guess so. I mean, that is true. We can't say whether they can go visit and there's nobody there to say, come in and translate me, and so they come back out.
Right.
I think we can't say that. Certainly by EM, we can see that the ribosomes are excluded. It's not like we see a few little ribosomes in there. It's remarkable. That's one of the ways by EM you see the granules the best, is that there's no ribosomes there.
Can you do FRAP?
For the ribosome.
Like you could bleach inside the, iFrap or something like that, where you show that there's diffusion in?
Of the ribosomes, you mean?
So,
I mean, they're so low level in the, I guess we could do that. We haven't done that. We haven't, yes.
So you showed that when you get rid of these scaffolds, degradation or translation are mostly okay, right? So what happens to the animals?
Exactly. They're actually fertile, which was the first thing that alerted to us that this is not that important. They're not 100% fertile. They're about 70% fertile. So we say the reason they're not fully fertile is because many mRNAs required for germline development are now in lower level. So for example, nanos, the favorite germline mRNA, is now lower level and there's less nanos protein. But this is really because of the localization function, not because of translation regulation.
So I have a question about the paranuclear way back here, perinuclear localization of the germ granules, are those directly connected to the nucleus via nuclear pores?
Yes, they are. So I just talked about the germ granules in this very specific stage of development where they're asymmetrically segregated, and I think the major function is to localize RNA. Later on, these granules become beautifully perinuclear, and they associate with nuclear pores, and lots of argonauts and small RNA machinery enriched in the granules, and it's thought that they might be specialized compartments for triaging mRNAs that come out of the nucleus, etc. Except we did the experiment and removed the granules from the periphery by messing around with nucleoporins, FG nups, and the worms are fertile. Okay, there are subtle changes to their transcriptome, and I can talk to you about that, but it's not, even that is not as important as we thought.
So I just would like to hear your comment on, so what exactly is nucleating these condensate? Is it like one single protein that's deriving this, or is it a combination of multiple interaction?
Yes, so I didn't go into the details. I mean, certainly MEG3 on its own can condense RNAs in vitro, and it's the granules, the P granules are actually MEG3 condensates on the surface of a liquid phase made by other proteins. So yes, it's complicated. There's multiple proteins that contribute, but in terms of the poly-A mRNA condensation, it's all MEG3 driven, because when we get rid of MEG3 or MEG4, the RNAs are no longer condensed. So P-bodies, that again is more complicated. So that was well established by Roy Parker and other colleagues, that the EDCs are part of the scaffold, so those are the ones that mutated. But indeed, when we get rid of EDC3 or EDC4, we don't obliterate P bodies. We do reduce the mRNA condensation in P bodies, but some proteins stay, some proteins go away. So again, it's a multi-component condensate, yes.
So just curiosity, so do you think it's so it's essentially the concentration of the RMP, so the decapping protein essentially, are those the only protein that will saturate and then transition, so forming like P body? Or there should be other RNA binding proteins over there?
Oh, so do I know that just the decapping,
yes.
What I drew there, is that sufficient to phase separate? Well, the Gross lab in vitro has shown that DCP1 and DCP2 can form condensates on capped mRNAs, yes? So at least in vitro, those two proteins are sufficient. And in fact, in his experiment, he suggested that when they are condensed, they're less active at decapping, and that this would go along with the suggestion that came that P-bodies are actually not sites of RNA decay, but more sites of RNA storage, okay? If that were the case, then maybe we would see faster degradation if we get rid of the P-bodies. We don't see that, although a few mRNAs do appear to be faster degraded, so we'll follow up on that.
I see there will be many discussions at the cocktail. So, given
Thank you
the
very
interest
much.
of time, thank you very much.